So at the BSH meeting this year, I'm presenting on a trial we call ENABLE. This is a phase 1 first-in-human trial of a new CAR T-cell construct that's a so-called third-generation construct. So this means it chains 2 different co-stimulatory domains together. And what's unique about the construct that we're working with, WZTL002, is that it links together a CD28 co-stimulatory domain with a toll-like receptor 2 co-stimulatory domain. And very few groups are working with that type of co-stimulatory domain. What we've seen in vitro is that when we add that TLR2 domain, we can get, under some circumstances, improved control of tumor cell growth, but also it modulates the cytokine profile. And in particular, it seems to result in reduced levels of a couple of cytokines, such as interferon gamma and GM-CSF, that have been linked to neurotoxicity, which is a particular type of toxicity often linked particularly with CD28 CAR T-cell therapies. What we found in the trial so far, and we've treated 21 patients in the dose escalation study, is that we've seen the toxicity profile overall is quite similar to what we might expect to see with other CAR T-cell products. But the biggest surprise to us, given that our product contains a CD28 domain, is an absence of neurotoxicity. So we've seen so far, fingers crossed, no neurotoxicity of any grade among the 21 patients treated so far. And this fits with some of our translational studies where we are seeing this modulated cytokine profile. We're looking at this in further detail, and we're planning to expand our cohort. In fact, we're currently enrolling to a dose expansion cohort to gain more information about this before we move on to automate our manufacturing and hopefully develop a CAR T-cell product that may have some advantages in terms of tolerability over certain existing constructs. So in the ENABLE trial to date, as well as having very low rates of neurotoxicity, we've seen no severe cytokine release syndrome, so only grades 1 and 2 CRS. We've seen cytopenias, as one might expect for CAR T-cell therapies, and we've had a very encouraging response rate with just over 50% complete response rate, comparable, I would say, to other second-generation constructs. And we look forward to finding out how these results pan out when we treat a larger number of patients.